Now we start video 2, which is the chapter 3 of homeostasis of A2 biology. In this we're going to discuss ultrafiltration, selective reabsorption, the structure and functions of the glomerulus, the PCT, the loop of Henle, the collecting duct and ADH, and I hope I can finish this in this video. Now when we look at the structure of the glomerulus as mentioned earlier in the previous video, this is called the afferent arteriole. So afferent arteriole, and then we have the one which is leaving it. So this is the one which is entering it, and this is the one which is leaving it. Now this will be called E for afferent, E for exit, efferent arteriole. Now in between we have capillaries, and you know the glomerulus, the capillary wall is called the endothelium. The first cell layer is the lining or the endothelium of the capillary, and the endothelium here has more gaps than in any other capillary in the body. Now what is supporting this one cell thick capillary wall is a basement membrane, which I am drawing in black. So this basement membrane is supporting the one cell. So everything can move out of the capillaries, but what is preventing it to move out is the basement membrane. So the basement membrane is made up of a network of collagen and glycoproteins. So the first we have the dotted line which I have shown to you here is the endothelium. Now this is, of course, we have studied this in the AS level biology. So this is called the endothelium, this dotted line which I have shown you. So these are the endothelium of the capillary, and then it is supported by the basement membrane. And then, of course, I am going to change the color. There is another layer of cells here which I am drawing with a squiggly line. And this layer of cells, which is also called the visceral layer of the Bowman's capsule. Now this is made up of cells which have a very special structure, and it's called podocytes. And it's like I always compare it like a catcher which we tie up our hair with. And then it continues like this. Now this structure is different. These are flattened cells. So that layer is called the visceral layer, and this is called the parietal layer. And then, of course, this is going to go into the PCT. So this part is here where the PCT starts. So this is the layer which is made up of the podocytes, which have finger-like processes which are sort of going to wrap around the capillary. But this is also called the visceral layer of the Bowman's capsule. Visceral layer of the Bowman's capsule. And this layer, which I have drawn with these flattened cells, because they basically have another function to perform. They just have to help the filtrate as the blood filters out. This just has to guide that filtrate down here. So this, out of the plasma which is going to filter out, now that is not going to be called the plasma anymore. This is going to be called, what is here is going to be called the glomerular filtrate. Glomerular filtrate. So glomerular filtrate is this liquid which is going to come down here, come down here, come down here, and all these sides, and then it's going to flow into the PCT. Now this whole process is called ultrafiltration. Ultrafiltration because the fluid which was inside the blood vessels was called plasma. Now, the fluid which is outside is called the glomerular filtrate. Now, what cannot filter out is the blood cells cannot filter out. They're too large. And the plasma proteins cannot filter out. But please remember, the thing which is holding them back is this layer which is called the basement membrane. So the basement membrane is actually, if we examine somebody's urine and we find proteins in that, we know that the basement membrane is damaged, because the capillary, the holes are large enough for everything to pass out, but the basement membrane is preventing anything passing out. So if the basement membrane is damaged, that happens in a disease called glomerulonephritis. So the process here which I have just explained to you is ultrafiltration. And another thing which you have to keep in mind is in ultrafiltration is that a back pressure is created. Why? Because this lumen is narrower, this lumen is larger, and there's also a sphincter muscle here which is going to further increase the back pressure, and this is going to help in all this filtering out. So the afferent arteriole is wider than the efferent arteriole, and this creates a back pressure, and this is going to help in the process of ultrafiltration. Now if you look at this diagram, now this page is going to explain to you what is going to happen in the PCT. Now PCT is the proximal convoluted tubule. Now as you can see here, this is the Bowman's capsule, and the glomerular filtrate is coming out, and this glomerular filtrate is going to flow into the PCT. Now the PCT has cells lining it. It's a tube, and the cells lining it have a very important function to play, because they're going to selectively reabsorb. So basically whatever is present in the glomerular filtrate first has to enter the cell, and then it has to pass down through the cell, and then it has to enter the blood. The blood is just going to carry it away. So please understand this, that the glomerular filtrate, whatever is present in the glomerular filtrate first has to enter this cell. Then it has to pass through the basal layer. So this side is called the basal layer, and this side is called the lumen side. Why? Because this is the lumen of the PCT. The PCT is a tube. The PCT is this tube which is going down like this. So it's a tube, it's got a lumen, and it's got another side which is called the basal side, which is the base of it. So this is the basal side, and then there is a basement membrane here which is actually holding these cells up, and the basement membrane is present everywhere. And whatever is absorbed is going to enter. First it is going to enter from, now this liquid falling from here is called the glomerular filtrate, and it has to enter the cell. So it's going to enter on both sides. I'm not going to say it's only enter one side, it's going to enter both sides, and then it is going to pass, and then it's going to go into the blood, and the blood is just going to carry it away. Now what we need to understand is that there is a very special type of a channel protein which is called the sodium-potassium pump. Now the sodium-potassium pumps are placed here. They're placed on the basal side. So there will be lots and lots of sodium-potassium pumps here, here. And what do? Now there's a very basic mnemonic which I ask you to remember, and that is Nokia. And this is 3 sodium out, 2 potassium in always. So 3 sodium out, 2 potassium in always. And the mnemonic for this is Nokia. It's easy for you to remember it. So the sodium-potassium pumps are placed on the basal side. I'm showing you these, green little blobs. So what is going to happen is this is going to create a sodium is going to be always low in this cell. Now sodium ions will always be low in the cell because we've got the sodium potassium pump pumping them out all the time. So sodium enters the cell and the sodium is pumped out. Now this is actually active transport. Now what is going to happen? Now in the glomerular filtrate, which is going to be flowing down here, we have a lot of sodium ions. So what we're going to understand is that the sodium ions in the glomerular filtrate are going to diffuse in, but you know ions cannot diffuse in, so there must be a very special channel protein here for this. So sodium ions are going to diffuse in. So what we need to understand is that sodium ions are going to, the sodium ions which are entering in the glomerular filtrate are going to move into the cell. So they're going to move on both sides. I'm not going to say only one side. So they're going to move here into this cell. But when they diffuse in, they actually bring with them glucose. So the glucose molecule is also brought along. So this is called co-transport. This is called co-transport. Similarly, amino acids will also be brought along with them. So amino acids will move in and glucose will move in. So basically the sodium is moving in first, and the sodium is moving in because the sodium potassium pumps are creating a low sodium concentration inside the cell. So less sodium in, sodium tries to diffuse into the cell. When it diffuses with it, it also brings along with it its glucose molecule. So the glucose molecule also moves in, and the amino acids are also brought in the same way. They diffuse in with the glucose. So the amino acids and the sodium ions, amino acids plus the sodium ions will move in. I think I just said something a little. So sodium, glucose both moving in, sodium and amino acids both moving in. So as the sodium ions move in and the amino acids move in, now this creates this a little bit for a short while is the concentration increases. So what is going to happen is water is going to follow. So water moves in by osmosis. But then what happens to the sodium and the glucose? The sodium and the glucose enter the blood. So the sodium enters the blood, the glucose enters the blood, the amino acids enter the blood, and all this is carried in the blood. So this is all going to be carried here in this. So the sodium and glucose ions are co-transported. Sodium ions, glucose ions co-transported. So sodium and amino acids co-transported, and then the water follows. Now this is a very good and a brief analysis of what happens here in the PCT, and just a repeat of what I just said. So this is of course the filtrate in the tubule lumen. So what we have here is the filtrate in the tubule lumen. We have this here. Then you can see here that the sodium potassium pump, so 3 sodium out, 2 potassium in, and that's the mnemonic for that is Nokia. And then we see how the sodium ions, number 2 as you can see here, number 2 is here, downhill sodium entry at the apical membrane, and number 3, then we have the reabsorption of organic nutrients and certain ions by co-transport at the apical membrane. The apical membrane is the one towards the lumen side, and this is called the basolateral membrane. So this side is called the basolateral membrane. And number 4, we have the reabsorption of water by osmosis through the aquaporins. And number 5, lipid soluble substances diffused by the transcellular route, and various ions and urea also diffuses by the paracellular route. So we have primary active transport, which is of course the sodium potassium pump. Then the secondary active transport is the one in which the sodium sort of moves in, is because it's secondary to the primary one. And we have the transport proteins and the ion channels and the aquaporins. So this is just a brief revision of what I just told you a little earlier. Now we come to the function of the loop of Henle. Now in the loop of Henle, as you can see, this is the filtrate. So this would be the PCT here, and this would be how the fluid is flowing down it, and this would be of course the DCT here. Now as you can see here in the PCT, this is the descending limb, and this of course is the ascending limb. And what we need to remember is that the ascending limb is impermeable to water. So we start with here, and I always call this 1 because this is the cause and then there's some effect. So number 1, sodium and chloride is actively transported out here in 1. So 1 sodium and chloride ions, active transport. And they are actively transported out into the area between the two limbs of the loop of Henle. So this area becomes highly concentrated. Now this causes the water to move out. So water moves out of the descending limb. Number 1, I said sodium and chloride ions moving out, so this area becomes concentrated. So water moves out of the descending limb. Now this, you look at here, 300, and now it's very highly concentrated. You can see it's 1200 here. As it rounds the bend here, now sodium and chloride is going to diffuse out. So here it's diffusion. Here water is moving out by osmosis. And here sodium and chloride is moving out by active transport. So first we've got to start here. We've got to start the story here, 1. Then 2, where water is going to move out by osmosis. Now of course the concentration becomes, as the fluid rounds the bend here, it's very, very highly concentrated. So sodium and chloride is going to diffuse out here. So this is the counter current multiplier, because you see this is the one which is going to cause the sodium and chloride to move out, and in the descending limb, so this is called the counter current multiplier. This is going to make it very important is that the water, as it moves out, then is going to enter the vessels around this, which are called the vasa recta, and the water is going to be carried away in the blood. So this place here, because please remember the loop of Henle is in the medulla. So this area is the medulla, and this area is going to remain concentrated because the water is going to be carried away in this blood vessel, which is called the vasa recta. And now we come to osmoregulation. And osmoregulation is the control of the water potential of body fluids. And this of course is a very important part of homeostasis, and what is involved is, number 1, the hypothalamus, the posterior pituitary gland. Please remember the pituitary gland is made of 2 components, anterior pituitary gland and the posterior pituitary gland. Anterior means in the front and posterior means at the back. So it is important that you remember this posterior pituitary gland. So the water potential of the blood is constantly monitored by specialized sensory neurons in the hypothalamus, known as osmoreceptors. So they, like we have thermoreceptors, so they are osmoreceptors. And these cells will detect a decrease in the water potential of the blood, because maybe there's too much of amino acids, there's too much of glucose in the blood. So then nerve impulses are sent along the neurons, where they terminate in the posterior pituitary gland. So the posterior pituitary gland then releases, please, I'm saying posterior pituitary gland releases, I didn't say produce, releases the hormone ADH. And ADH stands for anti-diuretic hormone. Diuresis means production of urine. So anti-diuretic hormone. That means somehow what is going to happen is that the water which was going to be passed out in the urine, that water molecule is going to be absorbed back into the blood. So the concentration of the blood is going to normalize. So anti-diuretic hormone. Diuresis means more urine being produced. Now if the anti-diuretic hormone is being released, it means that the blood is concentrated. So if the blood is concentrated, that means the water potential has decreased. So we need to reabsorb the water, which should not be lost in the urine. So water will be reabsorbed, and this water, when it is reabsorbed, will of course normalize the blood concentration. So please understand that osmoreceptors in the hypothalamus detect posterior pituitary releases ADH. More water is reabsorbed. We say it alters the permeability of the collecting duct. So basically the ADH is going to work on the collecting duct, and it is going to ensure that the water is reabsorbed. We have to realize that the ADH is traveling in the blood. So the hormone ADH reaches the collecting duct. It works on the collecting duct, so that will be the target organ. And what it has on the cell membrane of the cells of the collecting duct are signaling receptors. Receptors which are going to ensure that the signaling molecule gets attached to it. So it's got a complementary shape. So the ADH gets attached to the cells which are lining the collecting duct. Now this results in a number of series of changes and signaling cascade. And what is present inside the cells are aquaporins. And aquaporins are special channel proteins which were inside the cell. As soon as the ADH attaches to it, these vesicles move. These vesicles move to the lumen side. So this is the lumen side. And as they move towards the lumen side, they go and get attached. They will come and get attached to this part. So what we're going to find is, we're going to find these here. And these are going to come and get attached to, and what is going to happen? So the same vesicles which were present inside the cell now have moved and get attached here. And what do we have here? We have the water molecule here. So the water molecule which is going to be passed out in the urine, that water molecule is going to enter through this aquaporin, enter the cell, and enter the blood. So finally water molecules will, lots and lots of water molecules will enter the cell and then will move into the blood. So the blood concentration will be normalized. And because this is a short acting hormone, so this ADH has to be removed. So it will be removed. So the ADH is removed, and these channel proteins will move back into the cell. The channel proteins which were first attached here will move back into the cell and wait for again if the ADH is produced. So this we say alters the permeability of the collecting duct, and more water is absorbed and less water would be passed out in the urine. So urine will be less in volume. And why would it be concentrated? Because now the same urea is in a smaller volume of water. So less in volume and concentrated. Now this is how the ADH works on the collecting duct. So it is the movement of the aquaporins which were found inside the cell. So the aquaporins which were found inside the cell would have moved to the lumen side, and this would have helped the water molecules to move in and then move into the blood. So more water will be reabsorbed, and this will of course normalize the concentration of the blood, because we said the water potential had decreased. So this is the function of ADH, and that finishes this video, and the rest of this chapter will continue in the third video which follows this video. Thank you.